might not yet that might not yet be experts in the field of chronobiology. I thought of giving a very short introduction about circadian rhythms, and then the focus will be on melatonin and basically what we know first from lab studies about the effects of light on melatonin. And you know, in lab studies you can control very much everything, so the confounding factors. So it's then interesting to see what happens in the field, and that's what I did during my PhD. I did several field studies to see whether what we know from the lab really applies also in the field. So we live in a rhythmic environment, and this is because basically the Earth, by rotating around its axis, produces this alternation between day and night and light and darkness. And so basically it was for almost every organism on Earth very beneficial to develop an internal timekeeping mechanism, the so-called circadian clock, because it's about 24 hours, the period of the circadian clock, like the period of the rotation of the Earth around its axis. And the circadian clock is situated in the brain, in the suprachiasmatic nucleus, SCN also called, of the hypothalamus above the optic chiasm. And the circadian clock has many functions. One of the main functions is really to interact with the external light dark cycle and to anticipate changes in the environment, because you can imagine if we are just reacting to changes in the environment, we are going to always be too late. So it's very good to be able to have an internal timekeeping mechanism that's telling us when we should, for example, change our physiology to be active or to be asleep. And one example that you might all know is the cortisol release. So we have high levels of cortisol in the morning. We have this typical peak in the morning, and but the levels of cortisol are rising when we are still asleep. And this is what I mean here by anticipating the changes in the environment. So the circadian clock knows they will come, we have to be active, so it starts releasing cortisol when we are still asleep, so that when we have to wake up and get up from bed, we have high levels of cortisol. Another important function of the circadian clock is to regulate various biological rhythms in our body. So let me have a laser pointer. So the master clock, as I said, is in the brain, but we have basically clocks in every cell in our body. And so the master clock is gonna coordinate all these rhythms in our body, and it goes from gene expression to more complex behavior like the sleep-wake cycle. And another important feature of the clock is that it uses light to drain to the external light cycle. This is a simple scheme. Reality is much more complex to show you how entrainment works. So we have the SCN in the brain, the master clock. It receives inputs from the outside about the daylight dark cycle, light dark cycle. So whether it's day or night, and it processes this information and then it regulates physiology and behavior accordingly. And the output per se can also feed back to the SCN and to the Zeitgeber, because, for example, we can also change our own light exposure with our behavior. If we close our eyes, we're not going to be exposed to light anymore. So now I'm going to talk already about melatonin, the darkness hormone. It's called like this because melatonin is released and secreted during the night. So we almost have no levels of melatonin during the day in our blood, and in the night we have high levels of melatonin. And this is a typical rhythm that you can observe in the melatonin levels. So you see in the evening, just before we go to bed, usually it's 2 to 3 hours before we sleep, you have melatonin levels starting rising. Then you will reach a peak around the middle of the night, and then the levels will descend in the morning. Melatonin is produced in the pineal gland, which receives direct inputs from the SCN about the external light dark cycle, whether it's day or night. And we are going to see soon that light directly suppresses melatonin. So melatonin has been studied a lot in chronobiology, and the reason for that is because melatonin has a very stable and robust 24-hour rhythm. So it's a very good candidate to be a marker to study circadian rhythms in humans. And additionally, it's also very easily measurable, for example, in saliva. So you can just ask participants to take saliva samples, and something that they can also do at home, like hourly, and then you can measure the melatonin levels in the lab. The only thing that you really need to consider is that melatonin needs to be assessed in dim light conditions, because, as I already said, light directly suppresses melatonin. So you can use then different points in this curve to have your phase marker to then assess and study circadian rhythms in the field. One that has been used a lot, and I also used in my studies, is dim light melatonin onset. So when the melatonin levels start rising in the evening, just because you don't need to keep the participants awake all night and measure all night to have the entire curve, but it is enough to have 6 to 7 samples hourly in the evening before they go to bed, and then you can determine dim light melatonin onset. Good. So you can imagine that this rhythm is just a typical rhythm, but it can differ a lot between individuals. So if you measure melatonin in the population, you will see something more like that. So you will have the rise in melatonin in the evening at different times. You also will have different amplitudes. So there is a big variability in our circadian rhythms. And this is what we call then phase of entrainment. So each individual establishes a stable phase relationship with the external light dark cycle. And this is their phase of entrainment. And this is also what we can define as chronotype. So chronotype is our biological preference to be active at certain times of day and to sleep at certain times of night. And we can measure chronotype with simple questionnaires. And a questionnaire that I've used a lot in my research is the Munich chronotype questionnaire developed by Till Ronenberg. And this questionnaire simply asks about sleep timing on work days and on work-free days separately. And then you can, for example, have someone sleeping between midnight and 8 a.m. Then you can calculate the midpoint of sleep. Usually, to assess chronotype, we assess the midpoint of sleep on free days, because we assume on free days we are free from any social commitment, and so we can really measure our truly biological phase of entrainment. And then you can plot these results from a big population. So these are data from Germany. And you see that the majority of the population has a midpoint of sleep between 4 and 5. So probably people that sleep between midnight and 8, midnight and 9 a.m. on a free day. But you see that there is also here a big variation in sleep time. So we'll have very early sleepers, early chronotypes, and very late sleepers, late chronotypes, and basically everything in between. Then now we can ask the question, how do melatonin and chronotype relate? Of course, they are related because they both happen at night. And so you see, it's not a surprise that the melatonin rhythm will be advanced in earlier chronotypes compared to late chronotypes, and that we find a very nice correlation between melatonin peak and chronotype. So this was the little introduction about circadian rhythms, chronotype, phase of entrainment, and also melatonin. And we know already that light is very important for entrainment, for our synchronization with the external light dark cycle. So now the next question, and now I'm going to present you some results from lab studies, is what do we know about melatonin and light? So what are the effects of light on melatonin? This is one of first studies that showed that light can directly suppress melatonin. So here you had just two conditions. Participants were exposed to light at night, so between 2 and 4 a.m. And they were exposed to 500 lux and to 2,500 lux. And you see, especially when the light is so the intensity of light is very high, that there is a clear suppression of melatonin. So the melatonin levels clearly are lower when we are exposed to light. These are just other conditions. So if you have light intensity is in between 500 lux and 2,500 lux. And with more recent studies, we also know that there is a highly inter-individual variability in our sensitivity to light. So you will have individuals that are very sensitive to light, and for them also very low intensities will cause a melatonin suppression. And you also have individuals that are not so sensitive to light, and then you need much more higher intensities to suppress melatonin in these individuals. So this is something to keep in mind. Sensitivity to light can be very individual, like we have many different circadian rhythms. So there is always a lot of inter-individual variability. Another important feature of light is that it can shift melatonin phase. So this is a typical so-called phase response curve. So in this experiment, you usually expose participants to light pulses at different times of day, and then you will see the day after whether their melatonin rhythm has shifted or any other circadian rhythm. So 0 here is the timing of the core body temperature minimum, which usually happens in the middle of the night. So if you expose someone to light in the early biological night, so in the evening and when they would start sleeping, there will be a delay in the melatonin rhythm up to 3 hours. If you expose individuals to light in the morning or in the late biological night, there will be an advance. And you can already see that the amount of delay is a bit bigger than the amount of advance. This is because it's easier for us to delay our rhythms compared to advance them. Other bit of information, melatonin suppression and phase shift are stronger for shorter wavelengths. So we know now that our clock is mostly sensitive to blue or green light, to shorter wavelengths, and that's what you can see here. So the amount of melatonin suppression is higher if you're exposed to blue light and green light compared to amber and red light, and also the amount of phase shift is bigger if you're exposed to blue and green light. This is the last bit of information. So prior light history also can influence melatonin suppression, so our sensitivity to light in the evening. So in this experiment, participants were asked to try to live in dim light for 1 week. So they were supposed to stay at home and not go outdoor. And if they were going outdoor, they had to wear sunglasses to decrease as much as possible their exposure to light. And in the bright week, they were asked to be outside at least 4 hours per day. And after this dim week and bright week, they did a melatonin suppression protocol. So they exposed participants to light at night. And you can see that after the dim light week, the suppression of melatonin was higher compared to the bright light week. So if we are not exposed to a lot of light during the day, which is actually what happens to most of us because we live out indoor, then we are more sensitive to light in the evening. This can also partially explain why we have so many late chronotypes and late sleepers in society now. Good, so that was a lot of information about the effects of light on melatonin that we know from lab studies. Now I'm gonna continue with field studies, and I'm gonna mainly present my studies that I conducted in the Netherlands when I was doing my PhD. So I'm going to present 3 studies. Study 1 and 2 were aimed at testing some light interventions to see whether what we know from the lab can be applied in the field to try to advance sleep and melatonin, especially in late chronotypes. Because late chronotypes are usually in disadvantage in our society, since our society often has early starting times for school and for jobs. And so you can imagine, and maybe you have experienced it, late chronotypes usually sleep too short during the week, and then they have to catch up for sleep on the weekends. So the idea here was to try to help, especially late chronotypes, to advance their sleep, to get more sleep. And we used this information that we have from the phase response curve. So we know that if we reduce evening light exposure, we should advance our rhythms. And if we increase morning light exposure, we should also advance our rhythms. And then in study 3, it was an observational study, so no intervention. And we wanted just to describe how melatonin and sleep vary depending on the weekly schedule, so between work days and work-free days, and depending on season. So I'll start with the first study. So we had 2 groups. Both groups had 14 days baseline, where we were measuring daily sleep with sleep diaries and actigraphy. And then every Friday, we assessed DILMO at home. So participants were taking saliva samples at home. And then after these 2 weeks, we had the control group wearing glasses with clear lenses to have no intervention, but still to have like a behavioral similar intervention. So they were also wearing glasses for 2 weeks. And the intervention group was wearing blue light blocking glasses that look like this. So they are orange and they filter out especially blue light. But they also reduce light intensity by 50%. And the timing of the intervention was based on, was individual and was based on each individual chronotype. So we were trying to cover the most sensitive part of the day for these delaying effects of light in the evening. So this is the main result of this study. We found that sleep onset, but only on work days, and melatonin onset advanced after the first week of intervention. So here I plotted the data compared to baseline. So it's the change in sleep onset and DILMO compared to baseline for the 2 groups. And you see for the control group, there was basically no change across the weeks of the study. But for the intervention group, there was a clear advance in their sleep onset on work days, mirrored by a clear advance in melatonin onset. This was true only for the first week. And for the second week, we didn't have any more this significant effect. Now, this is an interesting question that I haven't really answered yet. And unfortunately, there are not so many studies that really have an intervention with this light intervention that is longer than 1 week. So to try to understand this better, I looked into the single individual to see how they behave, because sometimes you get more information than just looking at averages. And these are the individuals from which we have melatonin data. And so you see clearly 3 groups. So you have the people who advance week by week, or at least stabilize their advanced phase in week 2. So that's what we were hoping for. Then you see people who advanced during the first week, but then delayed again in week 2. And then there were these 2 kind of outliers who didn't really advance, actually they delayed. And the only, sorry, I have to drink some water. The only information that I had to look at was light exposure, but I don't think we can conclude too much from here. So if you look at this first group, you don't see clearly differences in the light exposure between the 4 weeks of the protocol. And if you look at this group that delayed, you might see here a bit of more light exposure during week 2 compared to baseline and week 1. But whether this explains, so that they had a bit more light exposure in the evening, whether this explains this delayed phase, I'm not sure. So then we have the second study. So here the protocol is similar. We had also an intervention group and a control group, and they also had 2 weeks of 10 days of baseline. And sorry, I have to drink some more water. So I hope my voice will stay with me. So after the 10 days of baseline, we asked participants to simply open their curtains for 2 weeks. So that was a very simple intervention. And the control group was always sleeping with curtains closed. And again, we assessed sleep with sleep diaries and actigraphy. And we assessed DILMO every Friday at home. So here we didn't find any significant effect on sleep nor on DILMO. So that was a bit disappointing. And we just find a correlation, a positive correlation between the change in DILMO and the change in morning light exposure in the bedrooms. But you see that this correlation is also mainly driven by by just these 3 participants. So these are the ones who really had a big change in their morning bedroom light exposure. So we had sensor near the windows, and they also had a big advance in DILMO. So basically, my take is that here the intervention was not strong enough to really have a significant effect at the group level. And there are many explanations for that. Of course, one thing is in which direction your window is facing, east, west, but also, and that I realized after the end of the experiment, many people in the Netherlands use just white curtains that don't really make the room dark. So if you then sleep with open curtains, there's not going to be a big change in your light exposure. Here I just wanted to show you a couple of results from other studies, other field studies that also investigated light interventions to shift melatonin and sleep. So this study is very similar to the one I did with the blue light blocking glasses. So here also they had just 1 week of intervention, 1 dim week where participants were wearing the glasses, but additionally they were also dimming their lights at home as much as possible. And then they had 1 bright week where they were asked to turn on all the lights at home and of course not wear the blue light blocking glasses. And here you see also a clear earlier DILMO in the dim light conditions compared to the bright weak condition. So this experiment is very similar to the one that I did, but they maximize the intervention. And then we have this experiment, which is also similar to the to the curtains experiment. So here they used a wake-up light, which increases light in the room during the last 30 minutes of your sleep. So when you decide to wake up. So they had 3 conditions: either no change in light, or the light was reaching a maximum of 10 lux, or the light was reaching a maximum of 250 lux. And here you see that there was also no effect on DILMO. So there was absolutely no change in DILMO. And again, I think here the strength of the intervention was not strong enough. And also we have to consider that if we exposed individuals to more light when they are sleeping, the eyelids are closed, so less light will reach their retina and their brain. So maybe something like this would work better, so that you expose participants to light when they are awake in the morning. And in these experiments, participants were not only be exposed to morning light at home for 30 minutes, but they also had a sleep advances scheme. So it's a combination of two interventions. But what you see is that if you try to advance your sleep by 1 hour every 3 days was here in this experiment, and additionally you expose yourself to light every morning, you can really advance your melatonin and your sleep. Good, so these were the intervention studies. And I'm gonna show you now the last study. So this was an observational study. We collected data in summer around the 21st of June, so the day with the longest photo period, and around the 21st of December, the day with the shortest photo period. And the study site was Groningen in the north of the Netherlands. We also, again, collected data about sleep with sleep diary and actigraphy. And we collected data for 10 days. And we collected DILMO on a work day and on a work-free day. We had only participants with a regular working schedule because we were really interested in the differences between work days and work-free days. So in this case, no students were allowed. And very important, the data collected in summer were collected under daylight saving time, but the clock times that are reported now in all graphs are in standard time, meaning that in summer 1 hour was subtracted from the clock time. So if we start with the seasonal effects, first in this graph you can see how photoperiod drastically changes in the Netherlands. So in winter we have about 7.5 hours of light, and in summer about 17 hours of light. So it's already a big difference, and I'm going to show you later also the intensity of light is very different between seasons. And here you see the distribution of DILMO, sleep onset and sleep offset in the sample. And so what we found was that both DILMO and sleep were earlier in summer, and they were about 1 hour earlier, except for sleep onset, which was only 30 minutes earlier. Sleep duration was shorter in summer because of this difference in sleep onset. And the phase angle difference between DILMO and sleep onset was greater in summer. So how do we interpret this data? By mid-June, when we collected the data, all variables were advanced by exactly 1 hour, except for sleep onset. And we think that this is a combined effect of the imposed shift in social schedules, because of course we have to wake up 1 hour earlier in summer, and a seasonal effect because light intensities were 10 times stronger in summer, as you can see in this graph. And we know from previous studies that if we are exposed to a lot of light during the day, this will also induce a phase advance in our circadian rhythms. And I'm going to show you one study that has shown that clearly, and it's a very famous study in our community, the so-called camping study. So here basically participants were brought camping, and so they had so they were exposed to much higher light intensities during the day, as you can see here, and basically no light in the evening compared to when they were at home and they were exposed to artificial light in the evening. And so if you compare melatonin, the melatonin rhythm, and also sleep between these two conditions, you will see a clear advance of the melatonin rhythm under natural light. So when we are exposed to a lot of light, our rhythms tend to advance. And we also have some interesting data from Russia, where in the last years there is no daylight saving time anymore, And here you see that also in this case, melatonin is earlier in summer. So this is the dots indicate melatonin. And in this case was 30 minutes earlier in summer, and sleep onset was 10 minutes later in summer, which actually really supports our results, because sleep onset in our case was the only one not really advancing completely by 1 hour in summer. And so also in this case, sleep duration was shorter in summer. Good, I still have a couple of results. So now we looked at the seasonal effects on DILMO and sleep. And then the next question was, how do they differ between work days and work-free days? We have a lot of studies, starting with the studies from Cronenberg, showing that there is indeed a big difference in our sleep between work days and work-free days. As I already said, especially late chronotypes usually sleep very short on work days, and then sleep much longer and later on work-free days. But it wasn't really clear what happens to DILMO, whether also DILMO shifts that much over a weekend without any light intervention. And what we found was that indeed also DILMO shifts. So DILMO on work-free days is about 30 minutes later than on workday. And sleep onset and offset, as we already know, are also later on work-free days compared to work days. So sleep duration is longer on work-free days, that is known. And the phase angle difference between DILMO and sleep was greater on work-free days. So what was also interesting here to see was that the weekday effect, so that DILMO is later on work-free days, was stronger for late chronotypes, which is not so surprising if you think that late chronotypes also shift their sleep much later compared to early chronotypes. And so they will be exposed to light later in the day because they sleep in. And indeed, if I compare the very early chronotypes in my sample to the very late chronotypes in my sample, the very early have no shift in DILMO and sleep. So they sleep regularly, and so also their DILMO doesn't really shift between work days and work-free days. But if I look at the latest chronotypes, they have a very big shift, up to 45 minutes in DILMO between work days and work-free days. Good. So that's all what I wanted to tell you. So I'm gonna wrap up. So light intervention can be designed to shift melatonin sleep timing, but I think that we need more studies to really determine which duration and strength are needed for effective light interventions. For me, it would be very interesting to know if somehow we can compare the two light interventions, so light intervention in the evening to reduce light, blue light exposure especially, and the intervention in the morning to increase light exposure. So the question would be, can we somehow determine which intervention is more effective so that we can then tell individuals who need it, you rather reduce your evening light, or you rather expose yourself to more light in the morning? So that is for me still an open question, and exactly what intensities do we need and how long we need to have the light intervention and so on. And then I've shown you that melatonin and sleep timing also show weekly and seasonal changes independent of any light intervention. So this is also something to keep in mind and to study more, because when we look at seasonal changes, for example, we still have the problem with daylight saving time. That is a confounding factor. Soon, this might be not a problem anymore. So it's going to be interesting to look at seasonal effects without daylight saving time. But I also think it would be interesting to look at how melatonin, for example, changes around daylight saving time, because I've shown you that by mid-June, melatonin was, so we were completely adapted to the 1 hour imposed shift. But what happens, so how long do we need to adapt to this shift? That's, I think, still an open question. And in general, I think we need more field studies assessing light and different biological rhythms longitudinally and continuously. And this is, of course, a limitation for melatonin, because to assess melatonin, we need dim light conditions, which is per se an intervention. So we cannot really do it every day. But I know, and I'm sure there will be more possibilities in the future to really assess several circadian rhythms for a long time, and things are developing quickly. So with that, thank you for your attention. I'm sorry because of my voice failure. And I would like to thank Martha Merrow and Thomas Kanter, who are my supervisor during my PhD, and Till Ronenberg and Eva Wienerbeck, with whom I discussed this data a lot, and who were also involved in writing the papers. Thank you very much.
Brilliant, Giulia. Thank you so much for the talk. We already have some questions coming in. So the first question is, if brighter light in early morning advances the clock, why was the sleep onset delayed in your seasonal study relevant to summer?
Sleep onset in summer was actually earlier. So it's 1 hour earlier in summer compared to winter, if I understand correctly the question. But we have these confounding factors of daylight saving time, as I said, that imposes this advance of our sleep. But also we have seen that with a lot of light, we also have an earlier sleep onset or earlier melatonin rhythm. Okay. I'm not sure if it answers the question.
Joshua wants to know, or says, great talk, thanks. Can you predict what long-term changes there might be if you perform study 1 for, say, months? So for longer time, basically. Do you think there could eventually be long-term circadian misalignment?
So circadian misalignment is something that is always difficult to define. I don't know exactly what Joshua means, but so that's what I'm saying. We have always studies with just 1 week or maximum 2 weeks interventions, and then we don't know. And indeed, I would find it very interesting, because if you to look at long-term effects, because if you tell someone, well, to advance your sleep, you have to wear the blue light blocking glasses every evening, but for how long? Do you have to keep on wearing them? Are you gonna advance like forever, or are you gonna find a stable phase relationship? We don't know. So I don't know, I cannot answer the question.
So I'm just wondering, are there any studies out there, Giulia, that investigate this more long-term? I mean, not just, let's say, 1 week in summer and 1 in the winter, but, you know, throughout the year, maybe with a really small sample size, but then tracking really individuals or something like that?
I couldn't really find anything with light interventions, so with a long-term tracking, but I might have missed something. So yeah, I don't know, but if anyone knows of such studies, I would be interested to see them.
Okay, I also have a related question to that, actually, concerning individual sensitivity. So because you very nicely showed the different groups that you found, so when you don't average over the people but actually group by how they respond, did you actually relate this to their individual light sensitivity, or do you know any study that looks at that in the field?
Yeah, so in my case, I couldn't do it because I didn't do any melatonin suppression in my study. So I didn't know the sensitivity to light of my participants. And I think this is relatively new. So in that sense, also, when we look at light intervention, it's always, you look at the group and you see whether it worked or not. But since we know that there are these big inter-individual differences, maybe this explains indeed my results, and I don't know it. But it could be that the two that were delaying were just not sensitive to light that much, so it didn't really matter whether they were wearing the glasses or not. Yeah.
Okay, thanks. Then Marco wants to know, hi, thank you for the really interesting data. In the third study, did you account for the type of work that your patients were doing in considering the shift in DILMO? Was the shift due to social cues or to the different exposure to light sources during work days or work-free days?
So I don't exactly know what type of work they did, but they were mainly office workers. So I didn't have any shift worker or people with like weird working schedules. And so I think actually what really makes the difference is, well, it's the egg chicken problem, right? But I can imagine if you're a late chronotype, then and you sleep too short during the working week, then on work-free days you're gonna sleep longer and later, and then just because of that you're gonna be exposed to light later. You're gonna miss this morning light pulse that would advance your rhythm. So that is my explanation why I find these differences also in DILMO between workdays and work-free days.
Okay. Then Maidel says, very interesting talk, thanks. Could light intervention alone be effective in treating circadian rhythm sleeping disorders long-term? For example, delayed sleep phase disorder.
Yeah, there are definitely several studies that have looked in particular at delayed sleep phase disorder because they really need an intervention. And it works. So I think there are studies with both this type of intervention, either wearing the blue light blocking glasses in the evening or being exposed to blue light in the morning, and it works. But again, I don't think we know how long the intervention needs to be or how to stabilize the new phase of entrainment, let's say, so that people don't fall back when you stop the intervention.
Mm-hmm. So, I mean, that's sort of related to the next question by Werner Osterhaus. Great presentation, thanks. You state that more studies are needed to determine which duration and strength are needed for effective light interventions. What purpose should the intervention serve? What makes an intervention effective?
I think the intervention would be effective if you can stabilize a new phase of entrainment. So assume you have someone who sleeps too late, so who cannot fall asleep before 2 a.m. So you want to advance their rhythm so that they fall asleep at least at midnight, and then you want to stabilize that. I think that is an effective intervention, and I don't know if we know how to do it.
And then maybe even the question arises, when is it effective? So how many minutes would you advance so it actually in your daily life actually it's good for you? So that's an additional question that we actually don't know. Yeah. Fernando says, thanks for the talk. Did you find any differences in response to light exposure based on age or sex differences? Thanks.
So in terms of age, I didn't have a big distribution. There are studies showing that, of course, with older individuals, it's very different. So they are also, I think, less sensitive to light they become with aging. But in my case, I didn't really have a broad distribution. They were all young, healthy people. And in terms of sex difference, also, I haven't really found anything in my sample. I think so far I remember.
Okay, lots of questions, Giulia. I will ask a couple of more questions. So Karen Bieske wants to know what lamps were used for the light intervention, fluorescent lamps or LEDs, which luminous color?
So in my study, I didn't use lamps. So for the other studies, I would refer to the references that I had. So I think the one with the blue light was just this typical Philips blue light that you can buy, and but I don't know the details, unfortunately. No.
Okay, then Werner again has another, I think, important question. Are there other purposes than sleep disorder treatments which would require a different light exposure?
So something that has not to do with sleep, because I would tell you late chronotypes who don't necessarily have a sleep disorder. Well, good question. I have to think about it. There might be other.
Yeah, I'm not entirely sure whether he refers to basically non-sleep effects. I'm not sure. Yeah.
I mean, there are, for example, the seasonal depression that is also, or depression per se, that is also treated with light. So, but that's not really to shift necessarily sleep timing, but to use the properties of light to cure seasonal depression, for example. Maybe that answers a bit the question.
Okay, then let's finish with the last question from Chiao Baksh. Thanks for the talk. Is there a sleep disorder in totally blind people, or do they get cues from something else?
Yeah, totally blind people definitely have problems with entrainment. So they usually try to entrain them with social cues, but it also depends whether, what is totally blind. So whether they really don't get any information about the light, because sometimes they cannot see, but I think Manuel knows better than me, but they still get the information about the light, and so they are entrained because of that.
Yeah, I guess it depends really on where they're blind, because then they would still get signals from the retinal ganglion cells. Okay, so with this, I want to thank you again, Giulia, for presenting today. It was really interesting. If you have any more questions, you can also you can drop me a mail, and then maybe we can negotiate with Giulia. Otherwise, the recording will be available for you. I stop it now. And just a second.